Thanks everybody for coming. I'm going to talk about a piece of work that I started last December when I was asked to prepare a paper for the Max Planck Institute in Munich. And I'll explain that since then I've set up an expert group. And this morning I was corresponding with the World Health Organization Global Governance and Ethics Unit, because the World Health Organization is currently looking at the governance of genome editing, and they've acknowledged that there's a patent gap. They haven't looked at patents in their studies, so they're encouraging my expert group, and if any of you'd like to participate, you'd be very welcome, to inform them about why patents are part of the governance system that the WHO needs to be looking at for genome editing. So I started really from the position with my study last year that while there have been intense debates underway about the scientific and ethical implications of genome editing, there's been insufficient attention paid to the patenting issues of these new transformational technologies. And while acknowledging some of the excellent work that's been done in this area on genome editing, the wider scientific community outside patent law experts really need to consider urgently the implications of patenting these inventions, of course taking into account notions of ordre public and morality. And also, while genome editing has great potential in terms of its transformational role in healthcare and well-being in society, the granting of these patent rights, as you know, can have profound implications in terms of affordability and access, particularly for people living with chronic lifelong illnesses or preventable inherited medical conditions. So what I'm trying to do is encourage the scientific community to consider carefully the impact of granting patents on genome technologies and the need to balance rewards for inventorship with the implications for affordability and access and human rights that this group, of course, are well familiar with. So genome editing technologies, as I said, there's great potential for research and for society at large. It's a fast, efficient and precise, relatively inexpensive tool to modify the cells of any living organism. So this might be a genome editing technique for cells of the body of a living human being, and we call that somatic cell modification. So you could potentially cure patients of chronic lifelong illnesses through somatic genome editing. There's also the issue that I'm really going to focus on, which is to do with the editing of human embryos in order to modify the germline identity of human beings. And you can probably see where I'm going to go with this, with the patenting of inventions relating to the germline identity of human beings, eradicating heritable diseases in newborn babies and creating resistance to lifelong conditions. What I'm not going to talk about, but in my wider group, which is advising the WHO, we're also looking at non-human uses of genome editing, such as new plant varieties, which are disease resistant or higher crop yield, and animal uses. The Nuffield Council on Bioethics is looking at this, and as part of my expert group as well, about how genome editing can contribute positively to food security. So great potential, and let me now explain a little bit about what CRISPR genome editing entails. So there were earlier versions of genome editing through zinc finger talons and the talons methods, which were quite complicated and unpredictable. And one of the issues, which I won't bother discussing in detail here, is that even with CRISPR, there's a lot of concern about off-target events. So you edit part of the genome and it has unexpected off-target effects, which might only exhibit in 6 or 7 generations' time. So those risks are less now with genome editing through CRISPR-Cas9. And essentially what this is, to give CRISPR its full name, it's clustered regulatory interspaced short palindromic repeats. So it's repeating sections in the DNA. And when CRISPR is used in combination with Cas9, which is an associated protein, which is given the number 9, this can then use CRISPR, which originally, or in its natural state, CRISPR is an immunological defense system. So the way that CRISPR works in its natural state is that within a living organism, the CRISPR will remember a virus which has attacked the living organism. And then the Cas9 will go in and, relying on the CRISPR to identify what the virus which has been encountered before, the protein Cas9 will go in and attack and cut out this alien virus. So a simple analogy would be with a word processor document, whereby the author would search for, and that would be the CRISPR part, searching for the misspelt word, and then the Cas9 would go in and delete the typographical error, and then could then knock in the correct spelling, in my analogy with a word processor. So it's a great technology, and if you read some of the leading scientists in this area, like Jennifer Doudna, they point out just how simple it is for scientists to use this technology. So it has great potential, but it also, of course, raises critical public policy issues that I think as patent lawyers we should be interested in. And that's really what I'm going to be talking about today. So just to give you a little bit more background to the science, the human genome is contained in 23 pairs of chromosomes in a sequence of paired chemical bases, which are held together in long molecules of DNA that are present in almost all cells of living organisms. And in my example, it's the human genome. So the genome's a complete set of the genes with regions of the DNA molecules of varying length, and they're interspersed with non-coding regions that regulate when the genomes are expressed. So although two people will have similar sets of genomes, even identical twins will not have exactly the same genome. From time to time, inherited genomic variations will result in disease or confer on an individual a predisposition to a particular disease. And this usually comes about due to small changes in the genome. And those can be transmitted onto future generations. So these might be life-limiting conditions such as muscular dystrophy or cystic fibrosis. Genetic conditions are also significant causes of infertility or pregnancy loss or neonatal death. And Huntington's disease is a typical example of a single genome defect, which potentially could be corrected through CRISPR-Cas9. So I'll explain a little bit more about how the technique works in a minute, but before I do that, let me just explain who the leading inventors are in this field, because we'll be coming onto these people when we talk about the patent disputes which have arisen. So CRISPR, in combination with Cas9, first came to public attention in 2012 with the publication of this seminal paper in Science by Jennifer Doudna at the University of California, Berkeley, and Emmanuel Charpentier, who at the time was based in Sweden and is now director of the Max Planck Institute in Berlin. And in their science paper, together with their collaborators, they demonstrated that CRISPR-Cas9 can be used to cut and possibly which is important. They said it could possibly use to edit DNA in vitro. So they predicted that it could be used for editing human embryos in the way that I've just described. So CRISPR-Cas9, it was adapted from this naturally occurring genome editing bacteria, which is part of the immune system. So the bacterial immune system captures snippets of DNA, as I explained with my word processing analogy. It captures DNA from the invading virus, and it uses them to create DNA segments, which are known as these CRISPR arrays, which then can identify a virus in the future. And if you were wondering, yes, there is a lot of research going on now about using CRISPR in the COVID-19 response. There's a lot of work being done on using CRISPR to generate a T-cell response in COVID patients. So to put it another way, in nature, when humans become infected from a virus, their immune system will use the CRISPR DNA and they then encode an RNA recognition sequence, which has been retained from the previous virus attack by the same bacterium. And that RNA sequence then produces a mechanism by which it matches with the previously known virus, and the Cas9 goes in and snips out the bacteria. So now let's start to come on to the patents. This is from the original patent, which was first disclosed as an invention in the Science Journal. So this is a US patent document. So it was pre-AIA, so it was under the old interference proceedings rules. I'll explain a bit more about the case in a minute, but the diagram which comes from the original patent document is quite illustrative. So what we're seeing here is that with CRISPR-Cas9 genome editing, what's happening in a laboratory is that the bioscientists are able to create a small piece of this RNA, which is a short guide. So you can see in the patent document, it refers to the CRISPR RNA, which is going in and it's guiding and creating a small piece of the RNA, which can then target a sequence of the DNA in the genome, and the RNA binds then to the Cas so that the genome editing can take place. So as I've explained, this is potentially of great significance in terms of medical research, because right now there's work being done on whether particularly single genome disorders like cystic fibrosis, hemophilia, Huntington's disease, sickle cell anemia, they could effectively go in by a scientist and cut out the defective gene. So what we're seeing here, as I've explained, is the CRISPR RNA, and we see a double-strand target DNA, and the CRISPR RNA pairs with another short RNA, and then a trans-activating CRISPR RNA is activating an enzyme to a viral or to some other DNA sequence that's already been identified, and then the Cas9 is going in and is cutting out the defective genome. And at that point, of course, you can then knock out completely the genome which is expressing for the disease in question, or you can even knock in a healthy genome. So you can knock out or knock in. So they call this system by which the enzyme is cleaving and destroying an offending DNA. Okay, so that was kind of the sciencey bit behind this. You'll have probably heard about most of what I've been talking about through the wide media attention that was given to the case of Lulu and Nana, the two baby girls, the twins that were born in China. And this was in November 2018, when a Chinese researcher from the Southern University of Science and Technology in Shenzhen, Dr. He Jianqiu, revealed at the second International Summit on Human Genome Editing in Hong Kong that he'd gone ahead, despite like an unofficial moratorium amongst the bioscience community, that they weren't going to mess around with this technology until it was deemed safe because of the off-target effects that I've already described. But he went ahead and did this and implanted into female embryo, 2 female embryos of these twins, an edited gene. And essentially what he did was that he disabled a genetic pathway for HIV. And he claimed to have disabled a gene which is called CCR5, which encodes for a protein which usually allows HIV to enter the cells. So this was the paper that he attempted to publish. So it was submitted to a number of leading scientific journals for peer review. And in an act of solidarity, the scientific community refused to peer review his paper. So it was leaked to the MIT Technology Review, but was never peer reviewed. So if you like, there's kind of a soft law by the scientific community that they were going to self-regulate this and not allow this research to be published. This gene called CCR5 that he claimed to have knocked out and then sort of edited, actually is present in about 6 to 8% of the global population. So 60 to 8% of the global population are less likely than the rest of the world's population to contract HIV because of the defect in the CCR5 gene. So he mirrored this in Lulu and Nana in their embryos, and these 2 baby girls were subsequently born. The objection of the scientific community, apart from the fact that there was a generally accepted de facto moratorium on this type of clinical application, is that if the gene is disabled, the twin baby girls with CCR5 disabled are now thought likely to be more receptive to other types of illness, such as the common flu virus or the effects of West Nile virus. So this is an illustration, again, from UC Berkeley of some of the studies that have been undertaken to show that actually the results of this type of in vitro work can have profound implications for the people who are born with the mutated genes. So that brings me again to the patent disputes. We saw earlier the diagram, which was from the UC Berkeley patent. I said that this was a pre-AIA patent. So now we see what happened when it went to interference proceedings and subsequently to the Federal Circuit Courts of Appeals. So Jennifer Doudna and her collaborators at UC Berkeley were the named co-inventors in the 859 patent with a priority date of 25th of May 2012. That's when the Science Journal was published. There were then interference proceedings lodged by the Broad Institute. Now, the Broad Institute is MIT and Harvard and various others. So it's a research institute based on the East Coast. And the Broad Institute claimed that the foundational work that had been done on CRISPR technologies by UC Berkeley, that they claimed that what had been specified in the patent document, in the disclosure and the claims, referred to a very broad set of claims to all CRISPR-Cas9 systems in a eukaryotic state. And also, the claims were very broad in the fact that they were all directed to CRISPR systems in any environment. So at the USPTO, the Patent Trial and Appeals Board then found in favor of the junior party Broad and said that it was terminating interference proceedings by accepting the claims of Broad pertaining to the use of the eukaryotic cells, which were sufficiently distinct because of the specific language used in the claims from the broader language used in the Berkeley claims to the use in any environments. So what we think is going to happen when these claims are reassessed at the PTO is that there will be specific boundaries on which elements are granted in the Berkeley patent with restrictions on the claims, and which parts of the technology are specifically allowed for Broad. So there you can see that how this was. The Patent Trial and Appeal Board decision was affirmed by the Federal Circuit also. The same set of patents then came up this January at the EPO. You probably recall that there were oral proceedings in January. This was a slightly different case. This was about priority. So it was an Article 85(1) EPC objection. And as you probably remember, what had happened was that it was claiming the priority on base. It was a PCT application based on the earlier USPTO application. But what they had effectively done, what Broad had done, is that they had omitted one of the original applicants, so that it became an issue of the priority of the application. And the question before the Boards of Appeal in oral proceedings in January is, are A and B as applicants for the priority application, when that's the case, can person A alone then be the sole applicant in the subsequent application, because applicant B had basically gone and worked for somebody else. And you might remember that this was dealt with quite swiftly in the oral proceedings. They didn't even use the full 4 days allocated, because they said, well, there's an established case law at the EPO about what any person means under Article 87(1) EPC. So none of this so far is really about the kind of the morality issues of the patent. So it's kind of that I want to now turn my attention to. Okay. You probably know that a lot of my work is actually on TRIPS and international patent law. So I take as my starting point, of course, Article 27(2) of TRIPS about the permissive language that WTO members may exclude from patentability inventions where it's necessary to protect order public and morality. And I won't even bother referring to the biotech directive, but of course then this language is reflected in Article 6 of the biotech directive in particular, and as you know, subsequently in the implementing regulations to the EPC in Rule 28, which talks specifically about the fact that EPC European patents shall not be granted in respect of biotech inventions, in particular concerning, and I've highlighted paragraph B, processes for modifying the germline genetic identity of human beings. The patent examiners in the examining division of the EPO that I've been talking to about the types of patent claims that they've been receiving in genome editing technologies, they've said, yeah, but it's not just about Rule 28 and morality issues. It's also about what's set out in the claims, and that the examiners, of course, always look at Rule 29, that the claims shall define the matter for which protection is sought in terms of the technical features of the invention. And the reason I'm explaining this is because in the cases that we've been looking at, Broad and UC Berkeley, when I actually looked at the European patent document and the claims and compared the European patent application filed with the patents granted, what I started to see was that the claim language had been amended during EPO patent prosecution. So having spoken to the examining division at the EPO, the examiner said, well, this is perfectly normal that the examining division would do that. Having regard, of course, to Article 28, but also thinking about how you can then apply the principles in Article 28 on the limitations of what constitutes patentable subject matter by using the claim construction, having regard to Rule 29. It's not surprising that you see what the examiners are doing is that the examiners are asking the applicant to explicitly state that, of course, what is being claimed is a system of delivering, so it's a vector. So the claim language has been amended to specifically provide that it says, provided that the said use of the vector, the delivery system, is not a process for modifying the germline genetic identity of human beings. So for the examining division, this is just perfectly normal that's what they would do. They would use Rule 29 as a mechanism to apply the principles of exclusion in Rule 28. So I just want to move on and finish in time for us to have a nice discussion about all this. I'm going to show you in a minute that there's actually an SSRN paper where I've talked about all this, and I'll show you a slide with the title of that paper in a minute. But in the paper, I also explore how the issues of patenting are actually related to human rights, because that's something I'm interested in as well. And the international law on germline genetic identity is really underpinned by the Universal Declaration on Human Rights of 1948, which of course you're familiar with, and then is expressed explicitly in the Oviedo Convention of 1997, which in Article 13 is the only piece of international law that talks specifically about modifying the human genome. And it talks about the fact that this should only be done in order to undertake preventive, diagnostic, or therapeutic purposes, and only if the aim is not to introduce modification into the genome of subsequent generations. So some of the discussions that I've been engaged in, I'll explain to you about the National Institutes of Sciences, National Academies of Sciences in the US, and the Royal Society have been holding a series of public consultations about the global governance of heritable human germline identity work in general. And they've been talking a lot about this provision, about how this relates to sort of human rights principles. So really just to finish this, and then I'll conclude with just a few extra slides. In my paper, what I'm really concerned with is how this relates to the public interest. If these patents have been granted, I believe that they're part of the governance system and need to be taken into account by the expert group of the World Health Organization, for example, which is currently consulting on this. There's been a lot of discussion about the welfare of the future person. You know, to whom are the human rights accorded? Is it to the parents to decide this, or is it to the future person who's not yet born, who's likely to have implications if their genome is edited? And the disability groups, for example, have had made a lot of interventions about the fact that people living with particular disabilities might be edited out of future society. And what does that say about people living today with these types of disabilities? Secondly, do we want to practice and do we want to grant patents on this type of bioengineering technique, and should it be released into society? And finally, who should have access to gene editing technologies? Our colleague Aisling McMahon, of course, has been doing some excellent work on the broad institute licensing options for allowing access to the patents that I've been describing earlier. So it's an issue of pre-grant about how we put limitations on the claims, and its issue of licensing, post-grant issues as well. So this is the paper where I really talk about all these things. It's on SSRN at the moment, and I'll be submitting it to a journal quite soon. Just to explain how this fits into my bigger project, my kind of book project is really thinking about how we can break all these down into different components. So I think there are issues of risk. There are issues of how risk is regulated. For example, through the Human Fertilization and Embryo Authority in the UK, or equivalent bioethical regulators elsewhere. There are issues of ethics, so medical ethics specialists are involved with this, and Richard Ashcroft, my former colleague at Queen Mary, now at City University, and I talk a lot about this. I would argue that there are issues of rights and about who those rights are accorded to. Of course, I think there are issues of patents and how patents plays a role in the governance system. And as I've just explained, there are issues of access as well. And this is just a slide just to give you an example of what a complex picture we're dealing with here, because this is just to show you some of the different models for different types of pre-implantation or in vitro human germline genetic modification, how these are regulated in different countries. I think what's most interesting is in grey, you can see that a lot of countries don't have any regulation at all on these matters. However, these systems may well have recourse under national patent laws where they've used ordre public and morality provisions to take these into account. So that I think there are issues about how patents can play a role in systems which don't have other governance mechanisms at present. So I mentioned that there are a number of international initiatives that have been looking at this, and I talked about Dr. He and the CCR5 genome editing on Lulu and Nana. He gave that announcement in November 2018 at the second meeting of the US National Academies of Sciences, Engineering and Medicine, which in collaboration with the Royal Society has been undertaking a number of public hearings to prepare a report on this. So on the 3rd of September, just a few days ago, the report of the National Academies of Sciences and the Royal Society was published, and there was a launch event online. The report's, I think, 225 pages long. So straight after the launch event, I had a look to see whether there's anything about patents in there, and there's nothing about the patent system at all, which seems a bit strange in many ways. However, at the launch event and in the report, it says specifically that the International Commission have left matters of governance to the World Health Organization expert group, which was set up initially in August 2019. So that's why earlier this morning, I was talking to the Secretariat at the World Health Organization about how I think my expert group can inform the World Health Organization's deliberations on the governance of genome editing. So this is my last slide, and it's just to show you the amazing people who are on my working group at Queen Mary Intellectual Property Research Institute. Richard Ashcroft, I mentioned, who's a professor of medical ethics. Nick Bassel is the patent attorney who drafted the patent on Dolly the sheep and leads the team, the biology team at Kilburn and Strode. Peter Browder at King's College did much of the the groundbreaking work on pre-implantation analysis of human embryos. So it's great to have scientists who began by, I mean, Peter began by saying, this has got nothing to do with the work that we do. These patents have got nothing to do with what we do. And slowly, we've kind of encouraged the scientists in the committee to think about actually probably patents do have something to say for you. Ruth Fletcher, my colleague at Queen Mary, comes from a feminist law background. So it's really useful to hear, you know, about her work on, you know, the rights of pregnant women and the rights of the unborn child. Frances Flinter, again, is a geneticist working at Guy's and St Thomas's. Emanuela Gambini, I have to give a shout out to Emanuela, she's my postdoc, and she came up with a title addressing the patent gap, because we think there's a gap. Andy Greenfield at Harwell Institute in Oxford, he's doing groundbreaking work on animal husbandry and genome editing in animals. So again, it's a science perspective. Aisling, of course, you know, and I mentioned she's done great work on the Broad Institute patents and more broadly on the governance of in this area and the role that patents play. Peter Mills, Nuffield Council on Bioethics. If you haven't read their 2 reports in this area, I recommend them. And there's a Nuffield report coming out early next year on genome editing in animals. So that will be a series of 3. Timo Minson, my good friend, you'll no doubt know as well. Nora Schultz is from the German Ethics Council, and it's amazing to hear the different perspectives given the fairly recent history of eugenics in Germany, what a different approach a German Ethics Council takes to this. Peter Thompson is the chief executive of the HFEA, and it's really good to hear a regulator's view. And Jakob Vested and Esther van Zimmerman, you'll probably know already. They were part of Timo Minson's research team in Copenhagen, and they're both doing great work on the governance of patents as well. So that's kind of what I've done and what we're planning to do in the future. So I will stop and leave 20 minutes for discussion. Karen, over to you.